Greetings, everyone. Happy New Year. Thank you for joining us for another RRP hosted research webinar series. I'm Katrine Erickson, the Executive Director of the RUNX1 Research Program. Most of you, as I can tell from the names who are joining, are probably very familiar with RRP, but just in case some of you are new to our community, we are a nonprofit organization dedicated to supporting patients with RUNX1 familial platelet disorder, which, as many of you know, has a very high predisposition to hematologic malignancies. And at RRP, we have one goal, and that is to prevent cancer in our patients and improve their quality of life. We at RRP, we do this through funding research, through building and supporting our patient community, through facilitating collaboration across the sort of broader healthcare ecosystem to discover treatment interventions that, again, will hopefully in the very near future prevent blood cancer in RUNX1 FPD. I do want to acknowledge that our funding impact would not be as significant if it weren't for our wonderful grant co-funding partners. We have partnerships with Alex's Lemonade Stand Foundation, the Leukemia Lymphoma Society, the Mark Foundation for Cancer Research, Edward P. Evans Foundation, as well as most recently a collaboration with the NCI and NIAID. And without them, we would not have been able to commit now over $10 million in funds for RUNX1 FPD research. So incredibly grateful to our partners. If you're on this call and you are conducting RUNX1 FPD relevant research and you're interested in presenting your research findings on one of our webinars, please don't hesitate to reach out to me. I would love to hear from you. All right, so I want to, before I introduce our renowned speaker, I do want to quickly remind everyone about the process of this webinar. So basically, our speaker will present for the first 35 to 40 minutes, and then we'll have a Q&A discussion at the end of about 15 to 20 minutes. For any questions that you'd like us to discuss during the Q&A, please submit them through the Q&A icon and refrain from asking questions in the chat because it's hard to manage both the chat and the Q&A. But if you want to use the chat function to provide feedback or comments throughout the talk, you're more than welcome to do that. And so with that, I would like to introduce Dr. Lucy Godley, who is the Hospira Foundation Professor of Medicine and Professor of Human Genetics at the University of Chicago. Dr. Godley, as many of you know, is devoted to the care of patients and families diagnosed with hereditary cancer syndromes. And she's an expert in the treatment of hematologic malignancies. And her lab has a particular focus in understanding how epigenetic changes influence cancer initiation and progression. Dr. Godley has published numerous papers on RUNX1 FPD specifically, and we know has taken great care of many families as well. Recently, in collaboration with the Mark Foundation for Cancer Research, RRP and the Mark Foundation funded Dr. Godley for her research project, which is the title of this presentation as well. And it's entitled Delineating the Epigenetic Landscape and Pro-Inflammatory Gene Expression Program that is associated with RUNX1 FPD. So we are delighted to have you here with us, Dr. Godley, and I'm very much looking forward to hearing your presentation.
Well, thank you, Katrina and Catherine, for inviting me to give this presentation today. And thank you all for coming. I always like to begin with acknowledgements, and certainly in this case, I really want to thank my patients. I was studying epigenetics in the lab, very basic biology of cytosine modifications, and in the clinic, seeing my patients with blood cancers. And I met a couple of families that had this lifelong history of a low platelet count. And then I was seeing them, various members of the family with myeloid malignancies. And I thought that maybe they had an inherited RUNX1 mutation, and I proposed this to my attendings. I was quickly told that that is a rare condition And I would not be seeing that in my clinic, and to, not mention that anymore. And years went by, and the patients did well, fortunately. So when I became an attending, they were still my patients. And with family number one, when I was finally able to do what I wanted to do as an attending, I tested the family for germline RUNX1. And yes, they did have a germline RUNX1 mutation. Fast forward about 6 months, and the second family that I had suspected might have germline RUNX1 mutation came to see me, and I was able to test them. And yes, they had germline RUNX1. And I thought, gee, if I have 2 families with germline RUNX1 mutation in 6 months of my clinic, there's no way this can be as rare as what I was told. Because what's the likelihood that I would have a once in 30-year experience twice in 6 months? So I thought this must not be true, that this is such a rare condition. And this is what really prompted me to start working in inherited leukemia. It was absolutely my experience with those 2 families, both of whom had germline RUNX1 mutations. And so I really, really thank my patients, because I feel like they showed me something that was underappreciated in the field. And I think now it's pretty widely accepted that inherited predisposition to blood cancers is common. But we're going to focus on RUNX1 today. And so just here on the left, this is a thank you that the daughter of one of my patients gave to me. But again, I really thank them. In the lab, we have lots of people who are really very dedicated to this work. And in particular, yoga has done some of the work that you'll see, as well as Simona, who has done some of the work that you'll see. And Monsi and Taylor work with me and with David Wu, who's my co-chair for the Myeloid Malignancy Variant Creation Expert Panel. And I'll begin by talking about some of that work, because it has really affected the way I think about DNA variants of all sorts, in particular RUNX1. And then my colleagues, Anupriya Agarwal, Emery Bresnik, Paul Liu, and Nancy Speck, we work together to try to understand how inflammatory signaling promotes leukemogenesis, both for germline RUNX1 and for GATA2. And I really want to thank them for all their discussions, and they've also really influenced how I think. And of course, the funding, which in this case would not be possible without the funding Katrine mentioned, the RUNX1 Research Program and the Mark Foundation. So I want to begin with a little bit of background. And for me, I'll set the background in terms of the Myeloid Malignancy Variant Creation Expert Panel. So the idea is, if you have a clinical laboratory and you're sequencing individuals, you will come upon sequence variation in those individuals. And when you see that, how do you know if that variation is a problem or not? There are 28 curation rules that the American College of Medical Genetics and Genomics has broadly defined. The idea of those curation rules was that they would be specified gene by gene in a very laborious process. And so a few years ago, the American Society of Hematology decided that this was an important process, and they were going to sponsor a so-called variant curation expert panel. I was asked to co-lead that panel with David. And so we have built this panel of biocurators and experts, and it has been really a tremendous experience. If any of you out there really want to learn how to curate variants, I would really recommend that you volunteer on one of these expert panels. There are lots of them now. We obviously work on genes where DNA variants confer risk to myeloid malignancy. But there is nothing like learning these rules by doing. And the process has really, I think, transformed the way we look at DNA variants in our research lab. And so, although we're doing sort of work for the greater clinical purpose, you will get benefit to yourself by understanding variant creation very deeply. And so this has been really a tremendous experience for me and the members of my lab. And I really think the reason we're able to do a lot of the science that we're doing now is because of the work that we've done on this panel. So to summarize the way this works, clinical laboratories all over the world, when they detect DNA variants, they deposit those variants in a system called ClinVar, which is open access, and you can look at all the different DNA variants in any gene of interest that you have. And you can see how that clinical laboratory has classified the variant. The classifications are shown here on the left: pathogenic, likely pathogenic. Both of these I describe as deleterious, and they are clinically actionable. Then you have variants of uncertain significance. You're really not sure whether that DNA change makes a difference or not. And then you have likely benign and benign. So again, the clinical labs are using ACMG rules to classify the variants, but in general, those classification rules are not specified gene by gene, and they really were designed to do so. So our variant creation expert panel specifies the ACMG rules for genes of interest. And the gene that we started with was RUNX1. We are now working on curation rules for GATA2 and DDX41, and we hope to get to CEBP alpha, ETV6, and ANCHOR D26. So what happens is we pull variants out of ClinVar. We have a group of biocurators who curate the variants according to our RUNX1 specified rules. Those classifications get looked at by an expert panel. And when we all agree, we put our stamp of approval on that classification, and that classification gets the highest level designation in ClinVar. So if you see an FDA designation in ClinVar, it means that a variant creation expert panel has applied its gene-specific rules to that variant. And so we have been doing this for RUNX1 for a little bit of time now. In 2022, we deposited 325 RUNX1 variants into the ClinVar system. And right now you can see that it's really a minority of these RUNX1 variants that are deemed deleterious. So 7% are pathogenic, 5% likely pathogenic. A decent number are benign, either likely benign or truly benign, but half of them are VUSs. And that is a problem because the VUS really should either be not a problem, benign, or problematic, deleterious. And so we need to do functional testing or segregation in families to push these VUSs into either extreme. And so I'll talk about how we can do some of that in a little while. Now, the MMVSEP takes its rules very seriously. And so when some of you who are scientists out there, when you're publishing papers and you curate a RUNX1 variant, we are a bit of a watchdog to see if you are using those creation rules the way they were intended. And I think especially at the beginning, when the rules first came out, people were not as experienced using the rules, and sometimes they were not applied correctly. And some members of our MMVSEP take that very seriously, and they will, as they did here, write essentially a letter to the editor correcting the variant curation. I tend to encourage manuscript revision when I am a reviewer of papers, and I feel like the variant creation is not being done accurately. And I think as you're reading papers, be very, very careful about how DNA variants are being curated. So people should be paying attention to whether, in the case of RUNX1, it's autosomal dominant. So you only need one deleterious variant to give you risk. But other conditions are autosomal recessive. And I've seen many, many papers where heterozygous deleterious variants in autosomal recessive genes are being counted. And of course, if it's autosomal recessive, by definition, you need two DNA variants, and you really should show that those are in trans, that they're not on the same chromosome. And so this MMVSEP does take its rules very seriously, and they will call out when they feel their rules are not being applied correctly. I would say over time, we're very pleased that more recent papers have applied the rules correctly. I think that's just really a process of getting to know how to use these rules over time. But I would watch that closely. Now, we are an advocate for RUNX1 because we are curating those variants all the time. And you all, especially the genetic counselors, will be familiar with the idea that the American College of Medical Genetics and Genomics also has a list of what they call secondary findings. So this is a list of genes that if you're doing, say, a whole exome sequence or a whole genome sequence, you have a responsibility to that individual to analyze those genes because they are clinically actionable. And this list is growing, as you might imagine. It's on the order of 75 or so genes now. And RUNX1 is not on that clinically actionable list. So that means if there is a person, a child out there getting a whole exome sequence or whole genome sequence for whatever reason, RUNX1 is not being analyzed. And we did not think that that was correct. We think RUNX1 is important, that it matters if someone has a deleterious RUNX1 variant and has platelet dysfunction, mostly. I think that's what you can do the most of. You know, there's not much we can do about risk to myeloid malignancy, but we'll get to that too. But certainly the platelet dysfunction is something that I think that's very important to know about. And so we petitioned, we being the MMVSEP, petitioned to the American College of Medical Genetics and Genomics to include RUNX1 in the next iteration of this list. And I am sorry to tell you that RUNX1 was rejected from being a clinically actionable gene. And you can see the reason here. It was discussed and not included, as you see, despite a well-established gene phenotype relationship. And the reason it was rejected, there were three. Number one, there are limited data on penetrance and prevalence from genomically ascertained family or clinic-based cohorts. Now, I don't think that's true. And many of you, some of you on the call have been involved in those family and clinic-based cohorts and have data on this. And that those data were included in the application. So this reason number one was pretty upsetting. Number two, the need for confirmation of the germline nature of the variant, which requires a skin biopsy. And that potentially imposes a burden on clinicians and patients. Well, I was not pleased with this reason either, because our application specifically said that confirmation of a suspected germline variant could be done either by using DNA derived from cultured skin fibroblasts or by segregation in the family. It said that multiple times very clearly. And I feel like this is the American College of Medical Genetics and Genomics. If they think determining germline nature of a variant is cumbersome and burdensome, we have a problem. And maybe most upsetting was number three, a non-catastrophic clinical presentation. And those of you who are at the RUNX1 research program meeting in October and heard from our patients and family advocates about the catastrophic presentations in their families of platelet dysfunction, especially, might take exception to this item number three. And so this comes from the paper of the latest. You can see the reference here. And so the MMVSEP doesn't take rejection lightly, as you might imagine. And we wrote back to this secondary findings committee of the ACMG and rebutted each of these points. And so I'm pleased to say that RUNX1 is being reconsidered by the secondary findings committee as a clinically actionable gene. And I suspect the MMVSEP is not going to let this go away until RUNX1 is added to the list. Once we're finished with RUNX1, we would like to add GATA2 to this list for similar reasons and beyond. But it is a bit of a fight, I would say. The application was quite detailed, and despite that, we still had those reasons for rejection. So stay tuned, but I hope this will change soon. So now getting to the research, the question that I have is, when you have a germline mutation and every cell in your body has that change, has that mutated gene, in which cell types does it matter? I think when we think about platelet dysfunction, obviously this matters in the megakaryocyte. When we think about myeloid malignancy or hematopoietic malignancy in general, it's certainly going to matter to have mutated RUNX1 in the hematopoietic stem cell, where RUNX1 functions as a master transcription factor. But are there other cell types in the body where this gene mutation matters? And the cell types that we really wondered about were within the bone marrow microenvironment. The stem cell does not sit in isolation, right? It sits against the bony trabeculi. It sits against the bone, and it's surrounded by nurse cells, if you will. And many of those are derived from these mesenchymal stromal cells. As you all know, the hematopoietic stem cell differentiates into its two main lineages, myeloid, lymphoid. We know that people with germline RUNX1 mutations are most at risk for myeloid malignancy, but they do get lymphoid malignancies, usually when they do their T cell malignancies. But people have these lymphocytes, floating around, circulating in their bodies for life. And we do have reports of autoimmune type diseases. And family number 2 that I mentioned at the beginning, the proband in that family had very severe eczema, which resolved after an allogeneic stem cell transplant. So I've always been very curious whether the lymphocytes in someone with germline RUNX1 is contributing to their phenotype or the symptoms that they have. So this is the basic question that this series of experiments is designed to answer. And like many of you, we have this idea that the inflammatory milieu in the body is promoting the transformation of this mutated hematopoietic stem cell. And this inflammation can come about in many different ways. So I just mentioned the eczema that I've seen in people with germline RUNX1. And obviously eczema is a condition where the skin is broken down, and that's an obvious entry point for all of the microbiota that are on the skin. In the lungs, you obviously have entry of airborne pathogens that can enter. And the gut, the intestines, is another source of where we have large number of bacteria and other microorganisms that can induce this inflammatory milieu. Because every cell in the body for somebody with germline RUNX1 has that mutation, then I wonder, these mesenchymal stromal cells themselves, are they pro-inflammatory? And is that contributing to the inflammation that the hematopoietic stem cell is seeing? And also these lymphocytes, are they themselves hyperinflammatory because they have this mutated RUNX1? So these are the underlying questions. So to start with the B cells, these are relatively easy to start with. Because through the years, we have made what I call cell products from our patients. So when they come in, they give us peripheral blood, they give us bone marrow aspirate. We make primary mesenchymal stromal cells from the bone marrow aspirate. And from the peripheral blood, we try to transform the lymphocytes using Epstein-Barr virus. And so we have lymphoblastoid cell lines from patients, say, as a control, germline DDX41, or in this case, germline RUNX1. The wild type cells are collected from normal people, and we buy them from Coriell. So these are commercially obtained LCLs. What we have done here with the LCLs, what you can do obviously is grow them under very defined conditions, because they're a cell line, and so they're very easy to grow. And so we grow them in parallel, the wild type DDX41 or RUNX1 mutants. And right now we have RNA-seq pending from those. Cytokine profiling is being done from conditioned media, and we're also measuring R-loop formation as another pro-inflammatory stimulus within the cell. So our hope is that we'll have this answer relatively soon. Now, the microenvironment, this idea that, again, the hematopoietic stem cell is sitting in the bone marrow surrounded by cells that are derived from these mesenchymal stromal cells. This study, which was done in germline CEBP alpha, I think is the strongest evidence that the microenvironment can affect the phenotype of the individual. So for germline CEBP alpha or CEBP alpha mutant AML, we know that there are different varieties of the AML, if you will. Most of the time, CEBP alpha mutant AML has 2 mutations. Each allele of the CEBP alpha is mutated. And most of the time, both of those are somatic. But about 10% of the time, one of those mutations, usually the 5'N mutation, is a germline mutation. You can also have single mutated CEBP alpha mutant AML, and that's not considered good prognosis. You can see that here. The green and the red are AMLs with 2 CEBP alpha mutations, and they have a better prognosis than the single allele mutant. So that's why we say biallelic CEBP alpha mutant AML, it has a good prognosis. Distinguishing the green from the red is the fact that in the green line, these are individuals where one of those 2 CEBP alpha mutations is inherited. And you can see that those individuals do the best. Now, in the leukemic stem cell, which is transformed, in both cases, green and red, you have 2 CEBP alpha mutations. But in the green, where you have the inherited mutation, every cell in that person's body has the CEBP alpha mutation. So in the bone marrow, every cell has the CEBP alpha mutation. And that is the best prognosis, suggesting that the surrounding cells in the bone marrow confer a chemosensitivity on the AML in these individuals. And so I have really maintained this question for each of these disorders: how much does the microenvironment contribute to the transformation in the first place, to chemosensitivity, and potentially even to relapse? And so, as I said, we do baseline bone marrow biopsies on these people. We do bone marrow aspirates, and it's pretty easy to grow these mesenchymal stromal cells from the bone marrow aspirates. And so we had done a lot of this work in the context of trying to understand how often a germline mutation caused myelodysplastic syndrome, and had grown out MSCs from many individuals who were diagnosed with MDS under the age of 40. And so we grew those MSCs originally just to isolate non-hematopoietic DNA. But once we had the cells, we had the cells. And once we did the genotyping, we knew which MSCs were actually mutant. And that allows you to ask whether those MSCs can differentiate normally when they have a variety of different germline mutations. And here on the right, you see all of the different primary MSCs that we did these kind of colony assays in. And the RUNX1 are here at the end. There are 3 of them. And you can see they're all, they fail to grow colonies compared to the healthy controls. So this suggests that the bone marrow microenvironment in a germline RUNX1 mutation carrying individual might not be normal, and might contribute to either the transformation of the hematopoietic stem cell, or the growth of that transformed stem cell, or the chemoresponsiveness of those leukemia cells. So we all think of RUNX1, I mean, we know RUNX1 as a master transcription factor is important in the hematopoietic stem cell, but is it actually expressed, or is it important in the mesenchymal stromal cell? So when we had all these MSCs, it was very easy to see if the gene was even expressed. This is just a slide to show you the process. The process is very easy. We take a bone marrow aspirate, we lyse the red cells and just plate. Now, for those of you in the research laboratory or in medical centers, this is the process by which cancer cytogenetics labs begin their cytogenetic analysis. And so what a cytogenetic lab will do is after 24 hours, they'll take the floating hematopoietic cells and do their either karyotyping or FISH or other studies, and they'll throw this flask in the garbage because the mesenchymal stromal cells have sat down on the plastic. It's sort of like a negative selection. So when we have a patient of interest, we'll often ask our cancer cytogenetics lab not to throw out the flask. And then we go and collect that flask. Now, the media that your cytogenetics lab is using is typically RPMI for hematopoietic cells. And your mesenchymal stromal cells are not going to be particularly happy in that media. So you do have to change the media pretty quickly. But it's a very easy way of making your MSCs. And we do this all the time. And this is how, as I said, we have these cell products from individuals. So when we took the MSCs from normal people, we asked, is RUNX1 expressed? And the answer is yes. So RUNX1 is expressed just like it is in a hematopoietic stem cell. We know from cleidocranial dysplasia that RUNX2 is very important in bone development, and RUNX2 is also expressed. RUNX3 is not really expressed very highly. So the first thing that we noticed when we took these RUNX1 mutant MSCs and put them in culture, you can keep the MSCs growing in culture as MSCs, as these omnipotent cells, or you can ask them to go down these differentiation pathways, depending on how you, on what media you use in the cells. So what we saw is that the RUNX1 mutant cells spontaneously differentiate into myofibroblasts. And this is smooth muscle actin staining that's consistent with myofibroblast differentiation. And you can essentially genotype MSCs by this phenomenon. We haven't seen this phenomenon for any other genotype. And obviously, when these cells have differentiated into myofibroblasts, they're not expressing some of the genes that are normally expressed when they're in their mesenchymal stromal cell state. And so that's the first observation. Obviously, when they're terminally differentiated as myofibroblasts, they won't grow in MSC colonies. And so you can see that here. But the problem with primary cells is the RUNX1 mutation is not the only difference among these different primary cells. So individuals have lots and lots of DNA variations that as we talked about at the beginning. And so how can I tell you that this phenomenon is due to the RUNX1 itself and not some other DNA variation that happens to be in the cell? And so we decided that a better way to really ask whether RUNX1 has an important function in MSC differentiation would be to make an allelic series And there is an immortalized human bone marrow derived MSC line. Actually, there are a couple of them. And the one that we use is able to go through differentiation down the different lineages. So it's very, very nice because it's a cell line and it was made from a certain person, but we can make an allelic series within that cell line where the only difference is the RUNX1 variant. And then we can ask whether differentiation is normal or not. And so we use a very standard CRISPR-Cas9 with HDR templating approach that many of you are probably doing in your labs. We make a polyclonal cell line. We then do single cell cloning, isolate the DNA from those single cells and sequence it. I'll share just a little bit of that with you. Now, when we make this allelic series, we need to have a series of positive controls and negative controls in order to potentially down the road test, say, a variant of uncertain significance. So what I'm going to show you is our attempt to make these positive controls. And these R201 and R204 are some of the more common of the germline RUNX1 variants. So we wanted to have those as positive controls. So these are just pictures of how the differentiation works. When you take the MSCs and put them down different differentiation media, they will go down whatever lineage you're asking them to go down. And then you can do special stains or gene expression studies to assess the degree of differentiation into adipocytes, chondrocytes, and osteocytes, for example. So we've done RNA-seq a little bit. We're going to do more of that, cytokine profiling, and again, measuring R loop formation. So this is the first run, if you will, of the CRISPR-Cas9 HDR. And it was designed around R204. And we actually got very efficient editing right in this region. And so here are the wild type cells. These are cells with a frame shift right at 202. This is a deletion, in-frame deletion from 202 to 204. And here is just a point mutation. And so Simona, when she was here, she did RNA-seq on these, on this allelic series. What's shown in red are genes that are overexpressed compared to wild type controls, and in blue are those that are less highly expressed in the mutant cells compared to the wild type cells. And you can see there are many different genes that are involved in inflammatory signaling. So even in this very simplified experiment, if you will, where you have a single cell line and an allelic series of RUNX1, you get a very complicated picture of derangement of inflammation. This is going to be layered on top of differentiation of those cells. So these are with the cells cultured as MSCs. We still have to do the differentiation into the different cell types and then see how gene expression changes as the cells differentiate. And a larger allelic series. So I just wanted to show you our latest iteration of trying to increase the number of RUNX1 variants in the allelic series and making variants that have been seen in people, which I think it's very important to me that the work that we're doing is relevant to what we see in patients. And so in this case, we essentially repeated the entire experiment, except down here at the end, in trying to screen a lot of single cell clones, we thought we want a fast way of doing it, because if you do the DNA sequence, you actually have to TA clone it, which gets very expensive and tedious. And so we wondered if we could develop a restriction enzyme assay, which is sort of a poor man's sequencing, right? Since a restriction enzyme cuts in a sequence specific manner. And so this is the restriction digestion map of the RUNX1 sequence that's around the R204. Here's the R204 here. You can see all these different enzymes cut. But when you have the R204X or stop codon, you actually lose a couple of restriction enzyme sites. And that turns out to be very convenient, because if you do a PCR amplification around this gene of interest, say in this case, we make a 244 base pair amplicon, it'll either be wild type or in theory, we'll have our desired mutation. And we cut with TAC1, we cut this amplicon with TAC1. If you're wild type, that cuts here and you get these fragments. If you are heterozygous with a mutation where this enzyme won't cut anymore, you don't cut and you retain that 244 base pair amplicon. And when we do this HDR, we either get homozygous or heterozygous. Those can be absolutely homozygous. You can get a compound het. So when you get the intact band, you still have to sequence it because it could be homozygous or compound het. So just to give you an example of what that looks like. Again, the schematic is up at the top, and then here are the controls. So wild type should cut and get 2 bands. The mutant should not cut at all, and it doesn't. And then these are just a few clones. So you can see in this case, you have heterozygous. So there's a mutant allele, and then there's a wild type allele that is mimicking what happens in people. And then you can even get something like this, which would appear either homozygous or compound heterozygous. And just to show you what you get, this is the sequencing results from that clone 4. This is almost a combination of the variants that I showed you earlier. This is the in-frame deletion from that deletes out amino acids 202 to 204, but it inserts a glycine. And so this is a little bit like what happens in people in terms of loss of 204, but the way this happens, you actually go right back into frame. So it's basically a surgical excision of R204, not quite. You have the glycine here, and then you're lacking 2 of these 3 amino acids. So this is the kind of thing that we're doing. We're screening lots and lots of clones to try to find ones that look like what we see in patients. And then the goal is to send these MSCs down different differentiation pathways, as I showed you, and measure the inflammatory milieu. Now, we have the hypothesis, again, that the inflammatory milieu provided by these mutated MSCs and potentially by the stem cells themselves are contributing to transformation of the hematopoietic stem cell. Now, many of you know about clonal hematopoiesis. For those of you who don't, I'll just very quickly say that as we age, all of our stem cells and all of our cells are accumulating DNA changes. And our sequencing capacity is so good now that we can actually measure this. And the blood is a very unique organ because the blood is really the only organ in the body where the stem cells, in this case from the blood system, put all their progeny into the same place and mix around in the peripheral blood. There's no other organ that mixes its progeny, right? So blood is really unique. So if you sequence blood, you're looking at the accumulated, and you see DNA changes, you're looking at the changes that are happening across the entire stem cell pool. And so if you sequence peripheral blood, you can see this process happening. You can see it in people who would appear to be healthy. And we now know this is itself a pro-inflammatory condition that gives risk for blood cancers, solid tumors, and probably lots of chronic conditions. We would really like to know if this process of clonal hematopoiesis, acquisition of somatic mutations, is a universal predictor of hereditary hematopoietic malignancies. And we and others, Paul and Anna and colleagues in South Australia, have been measuring clonal hematopoiesis in our patients. And we can observe clonal hematopoiesis very easily in germline RUNX1 mutation carriers, most commonly in the BCOR gene, which is not a gene that's typically mutated in, say, garden variety average person clonal hematopoiesis. So it really raises the question for me of whether there's something very particular in the bone marrow milieu that's allowing the propagation of cells that are germline RUNX1 mutant and then say have acquired a B-core mutation to promote the growth of those cells and potentially allow that cell to go on to give rise to malignancy. We don't actually know that the B-core mutated clonal hematopoiesis clone itself becomes the malignancy, or whether it's some sort of biomarker for, say, an inflamed state within the bone marrow. So that's something that we would also like to know. Is that clone itself the risk factor for developing disease? We would like to know if detection of these clones constitute molecular MDS. Is this essentially myelodysplastic syndrome where you're seeing it at the early stage? Why should we wait for overt dysplasia in the bone marrow or acquisition of other mutations in the bone marrow, which would define myelodysplastic syndrome? If we see an enlarging clone over time, is that enough to know that that person is at high risk for developing myeloid malignancy? And should somebody with a growing B-core clone get an allogeneic stem cell transplant before they even have MDS? These are some of the questions that we have. We don't have answers, but we're hoping that by combining our work on clonal hematopoiesis across laboratories across the world, we'll come to more of an understanding and really be able to help people think about their own individual risk and maybe even develop interventions to slow or stop that malignant progression. So that is where I'm going to end, and I hope I have given sort of something for everybody in terms of your background, and I'm happy to take questions now.
Okay, well, thank you, Lucy, for the presentation. And if people have questions, please submit your questions through the Q&A icon. So Lucy, I'm curious, you described some of the initial plans around or initial data around the gene expression that you saw. Can you tell us a little bit more about maybe some of those inflammatory genes that you picked up in the MSCs? And did you do the same experiment with the LCLs? And are you seeing patterns there? And how's that connecting to the epigenome?
Right. So we don't have the data back from the RNA-seq from the LCLs. I will be curious to see if any of the genes are the same, but I won't be surprised if they're different because the cells are grown. First of all, they're different cell types, right? One is MSCs, one is lymphocytes. They're grown in different media. One's grown on plastic, one are suspension. In the body, they're generally in different places, right? So I won't be surprised if there's not much overlap. I don't know if you noticed, I didn't make too much of a point of it, but in the inset to the figure of the RNA-seq that I showed you from the MSC allelic series, I was showing you each individual cell line. They're actually pretty different. So even such a subtle change around this, you know, the RUNX1 mutations that we compared were pretty similar to one another in a way. And the RNA-seq was pretty distinct for a lot of the genes. So what does that mean? I don't know. Also, these are growing MSCs alone in a plastic tissue culture dish. What happens in the people, right? Because in people, MSCs are not growing by themselves. They're in the whole milieu of the bone marrow. So I don't know how much what do we see in the tissue culture dish? How much is it really going to relate to what happens in vivo in the bone marrow? I don't know. So for me, all that is important at this point is that the cells don't look like wild type. They look different. We've tried to do staining on our patient material in the bone marrow to see if we see abnormal numbers of myofibroblasts, for example. Like we see spontaneous differentiation when we have the MSCs by themselves in the tissue culture dish. Does that happen in vivo? And to date, we haven't been able to see that phenomenon in people's bone marrow. So again, I don't know if, what you do in the lab is so reductionist because you're growing those cells in isolation on plastic. That's not what happens in vivo. So I don't even know if, how directly translatable our results from the lab will be. But, we'll see. I think, again, the important thing is just the general phenomenon of the inflammation. And what's really going to matter are in vivo measurements in people.
Do you plan on, you mentioned you had primary RUNX1 FPD MSCs. Do you plan on doing RNA-seq of those and then potentially comparing that to the cultured MRSC, or at least the cell lines that you're generating?
Yeah, exactly. We had that idea. The problem is they spontaneously differentiate into myofibroblasts and then they stop growing
Right away. So you can't get enough to do.
Well, we haven't gotten enough because we didn't realize it was going to happen. But now that we know that it happens, one could try to we also want to, potentially you could do single cell studies because that technology is much better now compared to when we started the work. Most single cell platforms are designed for physically small cells and the MSCs are very large. So I think we need to think about that as well. But I think that's the way to do it. Take a bone marrow aspirate out of the person and do single cell RNA-seq without any culturing, as fresh as you can, because I worry that there's so much artifact from what we do manipulating the cells in the lab.
Mm-hmm. Yeah. We have a question from a genetic counselor, and the question is, can a patient with BCOR, with a BCOR mutation, RUNX1FP patient, request a bone marrow transplant? What would be the data to reject that idea?
Yeah, so I think our patients are very educated, and our patients, our family advocates read the literature and they understand it. And so I have not to date had someone with germline RUNX1 watch their growing BCOR clone and ask to be transplanted, but it may have happened to one of my colleagues and it will happen soon because as we publish and talk about these ideas that, people are listening, right? The argument against doing a preemptive, by preemptive, I mean before malignancy transplant, is that the transplant itself has very serious risk. You have the risk of graft versus host disease, the risk of toxicity from the chemotherapy, potentially radiation is sometimes used at transplant. And so if someone doesn't have overt malignancy, most transplanters will be very, very hesitant to do a transplant without a malignant diagnosis. The reason I bring it up a little bit is, to push the ideas, right? To get us thinking about it. What does it mean to have myelodysplastic syndrome? That word dysplasia means a couple different things, right? It means technically funny looking cells under the microscope, but people have always assumed that they were malignant. Now that we have patients with germline mutations and pathologists are looking at these bone marrows when people do not have malignancy, they see dysplastic cells. They see funny looking cells. Now, sometimes pathologists are using the word atypical cells to distinguish them from dysplastic, meaning malignant cells. But I think even the language that we use can be complicated. When we say a cell is dysplastic, are we saying it just looks funny, or are we saying it's transformed and malignant? So this becomes complicated, and it's a bit of a slippery slope. So I think if you're a well-educated patient advocate and you have a growing B-Core clone, and maybe you have also a TET2 mutant clone, I mean, could you argue strongly that you would rather have an allogeneic stem cell transplant younger, when you are less likely to have severe graft versus host disease, before you have overt malignancy? I think you could make a pretty strong argument. If you went to enough transplant centers, I'm sure you would find a transplant center that would transplant you. But I say it right now to be a bit provocative. I think we have to be responsible, right? And that's why I say we don't know right now that B-Core mutant clone itself is going on to develop the malignancy. I only have one series of I think it was 9 samples collected over time, or 7 samples collected over 9 years in a single individual of a germline RUNX1 mutated patient who went from baseline to transformed disease. And Paul is sequencing that sample or series of samples. For the most part, we don't have that kind of serial sampling in an individual over time. And I think it's going to take those kind of studies to really understand, what does that B-Core mutated clone mean? Does it just mean someone at risk? Does it mean that is the precursor, that is the malignant cell growing? We just don't know.
Yeah. I mean, at least we know from the RUNX1 database and all the work that's been done by the international group of collaborators, is that the B-Core mutations are coming up both in the pre-malignant setting and in the malignant setting.
But they aren't from the same person.
But they're not from the same person, but there's definitely a pattern there.
Right, which is concerning.
Right.
It's concerning, right? It does, it's easy to say, easy to imagine that B-Core clone itself becomes the malignant clone, but we haven't actually shown it.
Even if it itself doesn't become the malignant clone, it's quite possible that it's contributing to an environment that selects for even more.
That's exactly right.
mutations.
Exactly right. Yes. And I think I was also asking, would family history make a difference? I mean, presumably if we're talking about someone with germline RUNX1, we're talking about a deleterious germline RUNX1 variant. And again, that variant curation is really, really important. So I don't actually think in this case, family history, I mean, maybe what you're getting at there is if there's a very high penetrance. I mean, I do think that not all mutations are created equal, and there might be some that are very, very highly penetrant. And then maybe if that family has a very highly penetrant allele, you might be more concerned. But again, I'd be careful, because maybe a family's highly penetrant because there are multiple germline mutations present, and multiple colleagues who are probably on the call here have those families where they don't just have germline RUNX1. I think Anna has a family with germline DDX41 and RUNX1. So, and we have lots of families with multiple germline mutations. So, is part of the penetrance in a family due to a combined effect, or is does that family have a very strongly penetrant allele? I mean, we'd have to look at the particulars in that family.
It's interesting. It's an interesting question. It's a complicated question because there's so many factors at play to consider. But we did have a discussion about this at the annual conference, not this previous year, but the year before. And it felt like most physicians on the call that were, used to sort of thinking about this and having discussions with patients, is that they all sort of said all of those things weigh into this sort of scorecard in their head.
Right.
Like, okay, there's a family history. Okay, most people in that family who were diagnosed with a malignancy, it happened in their 50s. So, they just sort of.
Yes, but remember, you have anticipation in these families, right?
Potentially, right. I don't know how strong the data is, but that's there too.
Should patients know their status? Well, I mean, my patients get all of their reports. So molecular profile, when a patient with a germline RUNX1 mutation has a bone marrow biopsy, presumably it's being done on a clinical basis. Molecular profiling would be a standard test now to be doing on that bone marrow biopsy. And yes, patients should have access to that. Can a saliva test come back negative incorrectly? So yes, saliva, in my opinion, is peripheral blood. Saliva is very, very highly contaminated with blood, and blood does this weird phenomenon of auto-correcting itself through somatic reversion. Not typically for RUNX1, but I think any gene can be reverted. And so I am a bit of a stickler in terms of diagnosis. That's why I advocate using DNA from cultured skin fibroblasts, because fibroblasts undergo somatic reversion much less frequently than hematopoietic tissue. So I would be surprised if a saliva test were negative, but in theory, it could be. If one presents with symptoms, should another test be done? I mean, definitely, you have to care for the person in front of you. And I'm always an advocate for very comprehensive testing. This can be hard for doctors or other healthcare providers who aren't ordering these tests frequently. But one of the families, well, the one I showed at the very end with the B Corps, I didn't go through the whole history of that family, but that family did not have comprehensive testing initially with their local doctor. The laboratory that did the test knew that a comprehensive test had not been ordered, and actually signaled on the report that further testing should be done. But because the local doctor wasn't familiar with the reports, the local doctor kind of missed that notation, because unfortunately, clinical labs often put that kind of notation in, 7-point font down at the bottom, and you have to have a microscope to see it. But fortunately for that family, the local doctor realized that he was confused about the report and called me and asked, am I interpreting this correctly? And I said, well, my biggest concern is that comprehensive testing wasn't done for the family. So let's get the proper comprehensive test done. And that's how the inherited deletion of RUNX1 was found. So deletion mutations where chunks of the chromosome are actually missing are relatively common for RUNX1. But unfortunately, doctors can order a test that doesn't cover deletions. And that's what happened to that family. So if a family has symptoms and a test is negative, you should definitely make sure it's comprehensive. Remember that not everything that looks and smells like a germline RUNX1 is. Germline ETV6, germline ANCAR D26, or what we call phenocopies. They present almost the same way. Typically, the platelet counts for ANCAR D26 are lower than RUNX1, but as we know that the platelet counts can be quite variable in these families. So I would say, you have to be an advocate for yourself and your family. If you're feeling like there's something there and it's not getting diagnosed, go to the expert who you have access to, people who do this every day and think about it every day, because the testing does have subtlety to it that the local doctors can have trouble with.
There's one more question. I know we're over time, but I thought if you have a few minutes, we can answer it, Lucy. Are you okay?
Yeah.
So the question is from a researcher, and it is as follows. Immortalized B cells are terminally differentiated B cells with overexpression of Epstein-Barr virus oncoproteins. Can these cells really reflect the stem cell characteristics?
Yeah, of course not. Everything is reductionist, right? Everything is imperfect. So I thought, like, for a first blush, at least all the cells are in the same differentiation state and can be cultured in a very defined way. When you start using primary cells, you're all over the map in terms of how the person's health is. Everybody's getting vaccinated now for COVID and flu, and that completely changes your lymphocyte profile. So there are other caveats to primary cells as well. So nothing is perfect, but it was the first blush of an experiment that at least was I could do in a very controlled way. And let's see what we get. And then, like you said, Katrine, we have to relay it to what happens in the primary cells over many people over a long time.
Mm-hmm. Great. Well, thank you very much for taking the time. And thank you all for your attendance and participation, and hope to see you on our next research webinar call.
Yeah, and email comments and questions. Thanks, everybody.
Thank you.
Bye-bye.
Bye-bye.